Coronavirus. Published by Wikipedia. Coronaviruses are a group of related viruses that cause diseases in mammals and birds. In humans, coronaviruses cause respiratory tract infections that can be mild, such as some cases of the common cold, and others that can be lethal, such as SARS, MERS, and COVID-19. Symptoms in other species vary. In chickens, they cause an upper respiratory tract disease, while in cows and pigs they cause diarrhea. There are yet to be vaccines or antiviral drugs to prevent or treat human coronavirus infections. Coronaviruses constitute the subfamily Orthocoronavirinae in the family Coronaviridae, order Nidovirales, and realm Riboviria. They are enveloped viruses with a positive sense single-stranded RNA genome and a nucleocapsid of helical symmetry. The genome size of coronaviruses ranges from approximately 27 to 34 kilobases, the largest among known RNA viruses. The name coronavirus is derived from the Latin corona, meaning crown or halo, which refers to the characteristic appearance reminiscent of a crown or a solar corona around the virions when viewed under two-dimensional transmission electron microscopy, due to the surface covering in club-shaped protein spikes. Discovery. Coronaviruses were first discovered in the late 1960s. The earliest ones discovered were an infectious bronchitis virus in chickens and two in human patients with the common cold. Other members of this family have since been identified, including SARS-CoV in 2003, HCOV-NL63 in 2004, HKU1 in 2005, MERS-CoV in 2012, and SARS-CoV-2 in 2019. Most of these have involved serious respiratory tract infections. Etymology. The name coronavirus is derived from Latin corona, meaning crown or wreath, itself a borrowing from Greek kovan, garland, wreath. The name refers to the characteristic appearance of virions by electron microscopy, which have a fringe of large, bulbous surface projections creating an image reminiscent of a crown or of a solar corona. This morphology is created by the viral spike peplumers, which are proteins on the surface of the virus. Morphology. Coronaviruses are large pleomorphic spherical particles with bulbous surface projections. The diameter of the virus particles is around 120 nanometers. The envelope of the virus in electron micrographs appears as a distinct pair of electron-dense shells. The viral envelope consists of a lipid bilayer where the membrane, envelope and spike structural proteins are anchored. A subset of coronaviruses also have a shorter spike-like surface protein called hemagglutinin esterase. Inside the envelope, there is the nucleocapsid, which is formed from multiple copies of the nucleocapsid protein, which are bound to the positive sense single-stranded RNA genome in a continuous beads on a string-type conformation. The genome size for coronaviruses ranges from approximately 27 to 34 kilobases. The lipid bilayer envelope, membrane proteins, and nucleocapsid protect the virus when it is outside the host cell. Replication. Infection begins when the virus enters the host organism and the spike protein attaches to its complementary host cell receptor. After attachment, a protease of the host cell cleaves and activates the receptor-attached spike protein. Depending on the host cell protease available, cleavage and activation allows cell entry through endocytosis or direct fusion of the viral envelope with the host membrane. On entry into the host cell, the virus particle is uncoated, and its genome enters the cell cytoplasm. The coronavirus RNA genome has a 5' methylated cap and a 3' polyadenylated tail, which allows the RNA to attach to the host cell's ribosome for translation. The host ribosome translates the initial overlapping open reading frame of the virus genome and forms a long polyprotein. The polyprotein has its own proteases which cleave the polyprotein into multiple non-structural proteins. A number of the non-structural proteins coalesce to form a multi-protein replicase transcriptase complex. The main replicase transcriptase protein is the RNA-dependent RNA polymerase. It is directly involved in the replication and transcription of RNA from an RNA strand. The other non-structural proteins in the complex assist in the replication and transcription process. The hexaribonuclease non-structural protein, for instance, provides extra fidelity to replication by providing a proofreading function which the RNA-dependent RNA polymerase lacks. One of the main functions of the complex is to replicate the viral genome. RDRP directly mediates the synthesis of negative sense genomic RNA from the positive sense genomic RNA. This is followed by the replication of positive sense genomic RNA from the negative sense genomic RNA. The other important function of the complex is to transcribe the viral genome. RDRP directly mediates the synthesis of negative sense subgenomic RNA molecules from the positive sense genomic RNA. This is followed by the transcription of these negative sense subgenomic RNA molecules to their corresponding positive sense mRNAs. The replicated positive sense genomic RNA becomes the genome of the progeny viruses. The mRNAs are gene transcripts of the last third of the virus genome after the initial overlapping reading frame. These mRNAs are translated by the host's ribosomes into the structural proteins and a number of accessory proteins. RNA translation occurs inside the endoplasmic reticulum. The viral structural proteins S, E, and M move along the secretory pathway into the Golgi intermediate compartment. There, the M proteins direct most protein-protein interactions required for assembly of viruses following its binding to the nucleocapsid. Progeny viruses are then released from the host cell by exocytosis through secretory vesicles. Transmission. Human-to-human transmission of coronaviruses is primarily thought to occur among close contacts via respiratory droplets generated by sneezing and coughing. The interaction of the coronavirus spike protein with its complement host cell receptor is central in determining the tissue tropism, infectivity, and species range of the virus. The SARS coronavirus, for example, infects human cells by attaching to the angiotensin-converting enzyme 2 receptor. Taxonomy. The scientific name for coronavirus is Orthocorona virini or Coronavirinae. Coronavirus belongs to the family of Coronaviridae. Genus, Alpha coronavirus. Species, human coronavirus 229E, human coronavirus NL63, Miniopterus bat coronavirus 1, Miniopterus bat coronavirus HKU8, porcine epidemic diarrhea virus, Rhinolephus bat coronavirus HKU2, Scotophilus bat coronavirus 512. Genus Beta coronavirus, type species, murine coronavirus. Species, Beta coronavirus 1, human coronavirus HKU1, murine coronavirus, Pipistrellus bat coronavirus HKU5, Rausetus bat coronavirus HKU9, severe acute respiratory syndrome related coronavirus, severe acute respiratory syndrome coronavirus 2, Tylonicteris bat coronavirus HKU4, Middle East respiratory syndrome related coronavirus, human coronavirus OC43, hedgehog coronavirus 1. Genus Gamma coronavirus, type species, infectious bronchitis virus. Species, Beluga whale coronavirus SW1, infectious bronchitis virus. Genus Delta coronavirus, type species, Bulbul coronavirus HKU11. Species, Bulbul coronavirus HKU11, porcine coronavirus HKU15. Evolution. The most recent common ancestor of all coronaviruses has been placed at around 8000 BCE. The mRCAs of the alpha coronavirus line has been placed at about 2400 BCE, the beta coronavirus line at 3300 BCE, the gamma coronavirus line at 2800 BCE, and the delta coronavirus line at about 3000 BCE. It appears that bats and birds, as warm-blooded flying vertebrates, are ideal hosts for the coronavirus gene source to fuel coronavirus evolution and dissemination. Bovine coronavirus and canine respiratory coronaviruses diverged from a common ancestor in 1951. Bovine coronavirus and human coronavirus OC43 diverged around the 1890s. Bovine coronavirus diverged from the equine coronavirus species at the end of the 18th century. The mRCA of human coronavirus OC43 has been dated to the 1950s. MERS-CoV, although related to several bat coronavirus species, appears to have diverged from these several centuries ago. The human coronavirus NL63 and a bat coronavirus shared an mRCA 563 to 822 years ago. The most closely related bat coronavirus and SARS-CoV diverged in 1986. A path of evolution of the SARS virus and keen relationship with bats have been proposed. The authors suggest that the coronaviruses have been co-evolved with bats for a long time, and the ancestors of SARS-CoV first infected the species of the genus Hippocidridae, subsequently spread to species of the Rhinolephidae and then to civets, and finally to humans. Alpaca coronavirus and human coronavirus 229E diverged before 1960. Human coronaviruses. Coronaviruses vary significantly in risk factor. Some can kill more than 30% of those infected, and some are relatively harmless, such as the common cold. Coronaviruses cause colds with major symptoms, such as fever and sore throat from swollen adenoids, occurring primarily in the winter and early spring seasons. Coronaviruses can cause pneumonia and bronchitis. The much-publicized human coronavirus discovered in 2003, SARS-CoV, which causes severe acute respiratory syndrome, has a unique pathogenesis because it causes both upper and lower respiratory tract infections. 7 strains of human coronaviruses are known. Human coronavirus 229E. Human coronavirus OC43. Severe acute respiratory syndrome coronavirus. Human coronavirus NL63. Human coronavirus HKU1. Middle East respiratory syndrome related coronavirus, previously known as novel coronavirus 2012 and HCOV-EAMC. Severe acute respiratory syndrome coronavirus 2, previously known as 2019 nCoV or novel coronavirus 2019. The coronaviruses HCOV-229E, NL63, OC43, and HKU1 continually circulate in the human population and cause respiratory infections in adults and children worldwide. Coronavirus survival times and transmission efficiency in warm and wet air. Studies have shown that warm and wet air lowers both the influenza virus survival times and the virus to human transmission efficiency. Airborne influenza virus survival may be less at air temperatures above 30 degrees Celsius where the humidity is above 50%. Virus survival times were much lower at higher temperatures and higher relative humidity. Virus-laden droplet nuclei are more efficiently produced at lower relative humidity because of increased evaporation of expelled droplet particles, such that more virus remains airborne longer. Do survive an infectious in air with a lower absolute humidity, such as less than 0.007 kg water per kilogram air. When airborne droplets evaporate, then very light airborne droplet nuclei are formed. Virus in airborne droplet nuclei may remain in the air for many hours.